So last class we have done our fifth class on S layers and its uses and functions. Also we talked about sheath, prostica, and fourth class we talked about capsule, right? And this is our fifth class, we talked about stalk. Today is our sixth class and we are going to talk about pili and fimbri. Many gram negative and some gram positive bacteria contain hair like appendages that are shorter, finer, and thinner than flagella and are used in attachment and conjugation rather than motility. So what happened, many gram negative bacteria and also some gram positive bacteria contain hair like appendages that are shorter, they are short, they are fine and thin than the flagella, and are used in attachment. They are not used in movement like flagella but used in attachment, to attached. Though they are almost same, some of them are historically called pillars, some of them are called fimbri. So some of them, pili and fimbri, are almost same, but some of them from the very first day are called pillars and some of them are called fimbri. Next, if we talk about pili, pili can be of two types. First is long conjugation pili or F pillars or sex pillars, and second is short attachment pili or fimbri. The second one is short attachment pili or fimbri. When we talk about long conjugation pili, that is F pillars or sex pillars, conjugation pili are genetically determined by conjugative plasmid. This conjugation pili are genetically determined by the conjugative plasmid. They help in conjugation, that is the process of transfer of genetic material from one bacteria to another. Okay, so they are transferring the genetic material from one bacteria to another, and this is called conjugation. So they follow conjugation and they had identified genetically by having their conjugative plasmid. Transfer of DNA generate genetic variety for bacteria. So the transfer of DNA, this generate genetic variety for the bacteria, and it is bad for human beings because it can pass antibiotic resistance. So if some bacteria is resistant to some antibiotic due to this kind of passage of genetic material, it can be there, and this will be very bad for human beings because if they attack the human beings and if the antibiotic is taken by the human being and it is resistant to the bacteria, then it will be really matlab, mortal, like. So it will be fatal, right? So yes, so this is the disadvantage. Next, we talk about short attachment pili. Short attachment pili. What is short attachment pili? It helps the bacteria to adhere to cell surfaces. Okay, same as that, but uh, they interface between air and water. So surfaces and interfaces. So it helps in bacteria to adhere to cell surfaces and also the interfaces that are between air and water. They increase pathogenically by enhancing bacteria to stick to surfaces. So they increase pathogenically by enhancing the bacteria to stick to surfaces. So pathogenicity increase this way. Now if you talk about example of Neisseria gonorrhoeae, the strain without pili does not cause gonorrhea, but pili contains cells are infectious because they stick to the epithelial cells of the urogenital system. So what happened? The Neisseria gonorrhoeae strain, which does not contain pili, does not is not infectious, but which is having pili is infectious because it stick to the epithelial cells of the urogenital system. They also stick to sperm cells uh, and transfer from one to another. Okay, E. coli contain pili that stick them in RBC. So the E. coli bacteria have pili which stick them to the red blood cell called hemagglutination. That they are also involved in forming biofilms. They are also involved in forming structures which are used in biofilm. Okay, so What do you mean by hemagglutination? Don't need to see anything. I have not written now anything on the diary. I will just dictate and explain, okay? Just like a classroom. So, what I'm saying to you is, what do you mean by hemagglutination? As I told, E. coli stick to the um, E. coli have pillars which stick to the RBC and cause hemagglutination. What is this hemagglutination? Hemagglutination, blood cell clump. This is called hemagglutination. So now you take your copy and make three columns. One is characteristic, second is pili, and third is fimbri. This three column we will write an answer. First is first we have written characteristics. So in characteristic number one is size. Characteristic number one is size. Pili. Under pili you will write larger than fimbri. Larger than fimbri. In bracket you will write 9 to 10 nanometer in diameter. 9 to 10 nanometer in diameter. 9 to 10 nanometer in diameter. Next, of Fimbri, same characteristic size is ongoing. Fimbri is shorter than Pili. Fimbri is shorter than Pili. Shorter than Pili. Shorter than Pili. Yes, in bracket you will write 3 to 10 nanometer in diameter. 3 to 10 nanometer in diameter. Next is next characteristic number 2 is number. Next characteristic number 2 is number. Less, only one or two per cell. In pili, under pili you are writing less, only one or two per cell. Number of pilis, characteristic number 2 we are writing less, only one or two per cell. Fimbri, under fimbri you are writing numerous, up to 1000 per cell. Numerous, up to 1000 per cell. Numerous, up to 1000 per cell. Third is formation. Third is formation. Governed by plasmid gene F plasmid. Formation under pili governed by plasmid gene F plasmid. Governed by plasmid gene F plasmid. And fimbri is governed by chromosomal genes. Governed by chromosomal genes. Governed by chromosomal genes. Function involved in cell to cell attachment during conjugation. Function involved in cell to cell attachment during conjugation. Pili function involved in cell to cell attachment during conjugation. Involved in cell to cell attachment during conjugation. Next is involved in cell to surface attachment of bacteria. Next is involved in cell to surface attachment of bacteria. Cell to surface attachment. Occurrence found mainly in gram negative bacteria. Pili found mainly in gram negative bacteria. Fimbri found in both gram positive and gram negative bacteria. Fimbri found in both gram negative and gram positive bacteria. So I hope so you all are hearing it and it's clear to you all. Okay. So this was your second class. Hope so you all understood it. And next class we are going to complete type 4 pili and hemi. Then we will move on with flagella, very important. And the last part of the outer structure. Then we'll move to cell wall and structure internal to cell wall. Thank you.